Imagine for a second just taking a swab of your own skin and not just using it to grow like a generic beating heart cell in a lab, but actually engineering the exact microscopic spark plug of the human heart.
Right. Highly specific tissue.
Exactly. And then taking it a step further to wire that custom spark plug directly into a functional nervous system. That is the mission of our deep dive today.
Yeah.
We are exploring a truly groundbreaking 2026 paper from the journal Cell Stem Cell by Zheng and colleagues.
Yeah, it's an incredible piece of work.
It really is. They have successfully grown a 3D, fully wired, miniature human biological pacemaker. And, we really want to unpack every single detail of this because it's just mind-blowing.
The scale of this is, it's difficult to overstate, honestly, because of all the historical roadblocks in cardiovascular medicine.
Right, because for decades, studying the heart's pacemaker basically meant relying on animal models, didn't it?
Yeah, exactly. Mostly mice. But a mouse's electrophysiology operates at, hundreds of beats per minute.
Yeah.
So it simply doesn't map perfectly onto human biology.
Because their hearts are just racing compared to ours.
Right. And then if you try studying human embryos to observe how our hearts actually wire themselves in real time, well, that's severely limited by tissue availability and ethical constraints.
Sure. And we've tried doing this in flat Petri dishes before, right? Like taking human pluripotent stem cells and kind of coaxing them into heart cells.
We have, yeah.
Yeah.
But those 2D models, they just hit a massive wall.
Because they lack the spatial architecture. It's like trying to understand the plumbing of a giant skyscraper by just looking at a flat piece of paper, a blueprint.
That's a great way to put it. The geometry matters immensely. I mean, in a real human heart, the physical spatial organization directly dictates the function.
Form follows function.
Exactly. And this is especially true for the sinoatrial node, or the CN.
Okay, let's unpack this for the listener. The SAN.
Yeah.
We think of the heart like a car engine. The SAN is the spark plug, right?
Yep. It sits right at the top of the right atrium. It literally initiates the electrical impulse for every single heartbeat.
But a 2D cell culture, the old way of doing it, that's basically having a spark plug just sitting alone on a workbench. I mean, it might spark, but it's not connected to the engine block, so it's not doing any mechanical work.
Exactly. So to fix this, the researchers had to figure out how to force these stem cells to grow into a 3D organoid, which they call a SANA.
Right, SANO for sinoatrial node organoid.
Right. But getting stem cells to commit to becoming this highly specialized pacemaker tissue, and not just generic heart muscle, that required a pretty massive chemical push.
And this is where they screened this huge library of compounds, right, looking for a trigger.
They did. And they landed on a chemical called 1-EBO, which functionally is a calcium-sensitive potassium channel activator.
Okay, wait, I want to pause on that mechanism because I know it's crucial for our deep dive today. Why on earth does opening a potassium channel tell a completely blank stem cell, hey, go become a pacemaker?
Yeah, it's wild. It all comes down to membrane voltage.
Voltage, like electricity.
Yeah, literally. By forcing those potassium channels open at a very specific stage of the cell's development, 1-EBO alters the electrical charge across the cell's outer membrane.
Oh, wow.
And this shift in voltage, it acts as an epigenetic trigger. It literally cascades down into the nucleus of the cell and turns on highly specific cardiac transcription factors.
So the electrical shift flips the genetic switch.
Precisely. And by adding 1-EBO, their success rate for creating these pacemaker cells shot up to about 50%.
That's huge compared to previous methods.
It is. And these cells confidently express the defining markers of the SAN, specifically two proteins you'll hear a lot about, SHOX2 and HCN4.
Ah, HCN4. This is the engine behind what's called the funny current. Right?
Yes, the famous funny current.
Which is just a great, objectively funny name for an ion channel. But it's called funny because it behaves completely backward compared to almost every other channel in the body, right?
That's right. Usually an ion channel opens when the cell's voltage spikes. But HCN4 opens when the voltage drops. So it allows this slow inward leak of positive ions that eventually builds up and triggers the next spontaneous heartbeat.
That is so cool. Okay, so before we move on, let's do a quick recap for you listening at home, because we promised to really drill down into these concepts. So far, we've got stem cells in a 3D environment. We hit them with 1-EBO, which opens potassium channels, that changes the voltage, flips the genes, and creates pacemaker cells pumping out SHOX2 and HCN4.
Perfect summary. But what's fascinating here is what happened after that 1-EBO trigger was pulled.
Right, because they didn't just stop there.
No, the cells didn't just express those markers and sit there in a random, disorganized clump. They actively self-organized.
Self-organized, meaning they just moved on their own.
Yeah, without any physical scaffolding, without any manipulation from the scientists, the organoid naturally divided into distinct geographical zones.
That is crazy.
They formed a head region, a tail region, and a transitional zone.
Wait, I'm stuck on this self-organization part. No one sculpted this tissue. How does a random microscopic cluster of stem cells know which end is supposed to be the head and which is the tail?
It's all about chemical gradients.
Gradients, okay, like a heat map.
Kinda, yeah. One side of the organoid just starts secreting slightly more of a specific signaling molecule, and that molecule diffuses across the cluster.
Oh, so the concentration gets weaker the further away you get.
Exactly. And the cells are constantly reading their physical position based on the local concentration of that molecule.
Wow, so they literally know where they are in the neighborhood.
Yes, and this creates a spatial map that perfectly mirrors the structure of an actual 16-week human fetal heart.
And you mentioned that transitional zone earlier. That's super important, right?
It is vitally important
Sure.
because an electrical spark from the pacemaker can't just slam directly into the regular working muscle of the atrium.
Why not?
It needs a bridge to step down the signal. If it hit full force, the electrical wave would just reflect back and cause a massive arrhythmia.
So it's like a buffer zone.
Exactly. And the organoid built that buffer zone entirely on its own.
Okay, so the architectural blueprint is intrinsically encoded in the cells once they get that initial push. But having a beautiful structure, yeah, and it's meaningless if we don't take it for a test drive.
Right, they had to prove it actually worked like a human heart under pressure.
So they ran some baseline pharmacological tests first. They administered a drug called ivabradine.
Which blocks that funny current we talked about.
Right, and it slowed the organoids beat down perfectly, and then they hit it with isoproterenol, which mimics adrenaline, and the beat sped up.
The basic mechanics were totally solid. But the real test, the thing that changes the game, was disease modeling.
Okay, here we go. This is where it gets wild.
They used CRISPR gene editing to actually insert a single specific mutation into the stem cells before they even grew the organoid.
And this was the KCNJ3 mutation, right? Specifically C.247AC.
Yes. And in humans, this exact mutation is responsible for familial sinus bradycardia.
Which is a condition where the patient has a dangerously slow, irregular heart rate.
Right. And to really understand the mechanics of why this mutation slows the heart down, we have to look at the cellular reset phase.
Okay.
It's called the maximum diastolic potential, or the MDP.
Let's use an analogy here for you listening. If a normal heartbeat is like a pendulum swinging back and forth, the MDP is the backswing, right?
Yes, great analogy. It's the absolute lowest voltage the cell hits before it swings forward again to trigger the next beat.
So the KCNJ3 mutation, it's what they call a gain-of-function mutation.
Right. It affects a specific potassium channel, but instead of just opening and closing normally like a door, the mutation basically props the door open permanently.
And because potassium ions carry a positive charge, all that positive energy is just constantly bleeding out of the cell.
Leaving the inside of the cell overly negative.
Which is hyperpolarization.
Exactly. Go back to your pendulum analogy. That constant positive leak pulls the pendulum way, way too far back.
Oh, I see.
So it takes significantly longer for the cell's voltage to climb out of that deep negative deficit and swing forward to fire the next beat.
Because it has so much further to travel.
Yes. So the overall heart rate just plummets.
And the mutant sanos in the lab, they exhibited this exact behavior.
They did. They beat incredibly slowly. But here's the best part. They didn't just observe the disease. They actively tried to rescue the tissue.
Using an experimental drug called tershipin Q.
Right. And tershipin Q specifically targets, binds to, and blocks that leaky mutant potassium channel.
It kicks the doorstop out.
Exactly. By plugging that leak, the cell's voltage stopped dropping into that extreme hyperpolarized state. The pendulum swing was corrected, and the normal heart rate was completely restored.
Okay, hold on. Let's do another quick recap because the implications here are staggering. If they can engineer a human genetic disease into this microscopic organoid using CRISPR, watch it fail, and then cure it, doesn't that mean we can completely bypass human drug trials for rare cardiac conditions?
It is a massive paradigm shift, yes.
I mean, you wouldn't have to risk giving an unproven experimental drug to a human patient suffering from bradycardia. You could literally just grow a petri dish version of their exact heart with their exact DNA and test the cure on a lab bench.
That's the dream.
Yeah.
We now have a high-fidelity platform to test highly specific, targeted therapies without any risk to human life.
But of course, there is a glaring limitation to just a standalone pacemaker, isn't there?
Yeah, there is. No matter how perfectly it beats, a pacemaker in complete isolation is blind.
Right. If you, the listener, go for a sprint right now, your muscles demand more oxygen and your heart needs to speed up to deliver it.
And if you fall asleep, it needs to slow down so it doesn't overwork.
And here's where it gets really interesting, going back to our car analogy.
Sure.
You know, having a perfect spark plug on a workbench, it still doesn't help us if we can't actively control the RPMs of the engine.
Right.
So the researchers, they didn't just stop at the spark plug. They grew the brake pedal, too.
They did. They needed to introduce the central nervous system. Because in the human body, the pacemaker doesn't just act alone. It receives its marching orders from a cluster of nerves called the right atrial ganglionated plexus.
The RAGP.
Right. So to recreate this, the team grew an entirely different structure from stem cells. They called it a CGPO.
Which stands for cardiac ganglionated plexus organoid.
Yes. And they packed this new organoid with parasympathetic autonomic neurons.
And parasympathetic, for our listeners, those are the rest and digest nerves, right? The ones primarily responsible for slowing the heart down.
Exactly. It's the brake pedal.
Which makes total sense because we know the RAGP sits right on the atrium in real life. So they took these two separate organoids, the pacemaker and the nerve cluster, and they fused them together.
They put them right next to each other, but they didn't just sit there side by side. They physically integrated.
Wow.
The main bodies of the nerve cells, they remained on the outside in their own cluster, but they grew these long, wire-like extensions called axons.
Basically living wires.
Yeah, and they grew directly into the sano. The axons physically wrapped themselves around the individual pacemaker cells.
Establishing a genuine neuromuscular junction?
In a dish, yes.
But wait, how do those axons know exactly where to grow? It's not like they have a tiny GPS map.
No, they don't. They follow a localized chemical scent trail.
Okay, kind of like the gradients we talked about earlier.
Exactly the same principle. The pacemaker cells were actively secreting a protein called BMP2.
BMP2.
And this protein acts as an act-on guidance cue. It creates a concentration gradient thickest right at the pacemaker and diffusing outward.
So the growing nerve fibers just sniff it out.
Basically, yeah. They simply follow the gradient up to its source, embedding themselves exactly where they need to be to control the beat.
That is just, it's almost spooky how smart these cells are. And they actually took this up another level, right? By adding a third component.
They did. An atrial cardiac organoid.
So just regular heart muscle.
Right, which created what they call the tri-assembloid.
So just to visualize this, you have the parasympathetic nerve organoid, which wires its axons into the pacemaker organoid, which then steps its electrical signal down into the atrial muscle organoid.
Yes, the complete three-part chain of command.
And this setup allowed them to manipulate the entire system dynamically, like playing a biological piano.
They used optogenetics for this, which is a really elegant technique.
Oh, I love optogenetics. Let's explain how this works. They genetically modify the neurons so they respond to light.
Right. So basically, a specific wavelength of blue light. So when the researchers flashed this blue light on the nerves,
the nerves fired.
They fired, and they released acetylcholine, which is the neurotransmitter that signals the pacemaker to brake.
And what happened to the muscle?
The electrical activity in the attached atrial muscle slowed down in perfect lockstep.
It hit the brakes perfectly.
It really did. And then, when they applied atropine, which is a drug that blocks those acetylcholine receptors,
it essentially cut the brake lines.
Exactly. The heart rate shot right back up to baseline, proving that the neural connection was the actual driving force behind the modulation.
They even tested thalidomide on this tri-assembloid, didn't they?
Yes, and that is a massive proof of concept for drug testing.
Because thalidomide is notoriously controversial. It's often used in cancer treatments today, but it has this bizarre, dangerous side effect of causing really slow heart rates in some patients.
And for a long time, the exact mechanism of that specific side effect was incredibly difficult to isolate in humans.
Right, because human biology is messy. There are a million variables.
But by exposing their isolated tri-assembloid to thalidomide, the team demonstrated clearly that the drug actually hijacks this exact neurocholinergic signaling pathway.
Meaning it acts on the nerves, not just the heart muscle itself.
Yes, it artificially amplifies that parasympathetic brake signal. And when they washed the thalidomide away from the dish, the baseline heart rate recovered.
Okay, recap number 3 for the listener. We've taken our custom spark plug, we've wired it to a living brake pedal using BMP2 gradients, and we've proven that we can speed it up and slow it down using light and drugs.
We have a fully functional system.
But there is a huge twist here. The nerves clearly act as a dynamic brake pedal. But that only explains what they do once the human is fully grown, right?
Right, in an adult state.
What were those nerves doing during the 40 weeks in the womb when this organoid, or a real human heart, was actually building itself?
This is arguably the biggest discovery in the paper. The researchers noticed that the timing of when these nerves invade the pacemaker during fetal development, it coincides perfectly with when the pacemaker cells stop multiplying and start maturing.
Oh, wow.
So they hypothesized that the nervous system wasn't just there to steer the ship, it was literally hoping to build it.
Okay, how do you even prove that?
They utilized a spatial transcriptomics technology called stereosec.
Okay, I really want to break down stereosec for a minute because the engineering behind it is just wild.
It is very cool.
Because traditionally, if you wanted to know what genes were active in a tissue, you'd take a sample, blend it up in a tube, and just look at the soup of RNA, right?
Yeah, bulk RNA sequencing. It tells you what's there, but not exactly where it came from in the tissue.
But stereosec maps the genes physically.
Exactly. You place the tissue onto a highly specialized chip, and this chip is covered in microscopic DNA barcodes.
Like barcodes at a grocery store.
Kind of. Every single microscopic spot on that chip has a unique barcode. So when you capture the RNA from the cells lying on top of it, each piece of RNA gets stamped with the barcode that corresponds to its exact XY coordinate on the chip.
Oh, I see. So you end up with a high-definition map showing exactly which genes are firing in which exact physical location.
Down to the cellular level.
That is incredible. So they used the stereosec on a real 12-week human embryonic heart, zooming in right on the border where the nerves meet the pacemaker.
And they intercepted a highly localized molecular exchange.
Like a secret chemical conversation.
Yes. They saw that the neurons were actively secreting a molecule called prosaposin, or PSAP.
PSAP.
And directly across the gap from them, the fetal pacemaker cells were suddenly expressing high levels of a highly specific receptor designed to catch it.
And this receptor is called GPR37.
Yes.
So just think about your own body for a second, developing in the womb. Your organs didn't just build themselves in isolated silos. Your nervous system was laying down a localized architectural blueprint, secreting PSAP to physically coach your heart on how to become a mature organ.
It's beautiful, really. If we connect this to the bigger picture of human biology, the behavior of GPR37 is a textbook example of developmental scaffolding.
Scaffolding, like on a construction site.
Exactly. This receptor is highly active in the embryonic stages while the heart is being built. But if you look at a fully grown adult heart, GPR37 is almost entirely absent.
So it exists purely to facilitate this one maturation window.
Right. Once the pacemaker is fully grown and mature, the scaffolding is dismantled and taken away.
Okay, but as we know in science, correlation isn't causation. Just because they are talking doesn't mean the talking causes the maturity. They needed to prove that this PSAP to GPR37 interaction was actually driving the maturation.
Right. So they did what scientists do best. They broke it to see what would happen.
Ah, knockout model.
Yes. They genetically engineered a batch of SANOs to completely lack the GPR37 receptor, a GPR37 knockout.
So the pacemaker cells are completely deaf to the PSAP signal.
Exactly. Then they fused these knockout pacemakers with the nerve organoids to see what would happen if that chemical exchange was blocked.
And let me guess, the pacemaker cells were completely chaotic.
Complete mess. They failed to mature on almost every single level.
Wow. How so?
Structurally, they couldn't build the dense collagen matrix that a mature node requires for its structural integrity.
So they were physically weak.
Yes. And electrically, they were chaotic. Instead of one dominant pacemaking site dictating a stable rhythmic beat, they had multiple competing initiation sites just firing randomly.
Like a band with 5 drummers all playing a different song.
Great analogy. And finally, they're calcium transients, which are the fundamental mechanism for how a muscle actually contracts. They were incredibly weak and disorganized.
Okay, so breaking the receptor ruins the pacemaker, but what if you go the other way? What if you take a normal Sino with its GPR37 receptors completely intact and you just flood it artificially with that PSAP protein?
Total transformation.
Really?
Yeah, the organoids just snapped into focus. They rapidly built the collagen matrix. They established a single powerful pacemaking site with synchronized, forceful calcium handling.
So it worked.
It absolutely worked. It confirmed, without a doubt, that the nervous system is the architect of the heart's maturity.
Okay, final recap before we wrap up today's deep dive. This research, it completely shatters the idea that we can study or engineer human tissues in isolation.
You really can't. Biology is too interconnected.
Right. By leveraging one EBO in stem cells, we can now grow a perfect 3D replica of the human pacemaker. By introducing chemical gradients like BMP2, we can coax autonomic nerves to wire directly into that tissue, which allows us to model genetic mutations like bradycardia and observe complex drug interactions without putting a single patient at risk.
Right.
And then, through the magic of StereoSec, we've uncovered the hidden PSAP-GPR37 pathway, proving that our nervous system actively coaches our heart into maturity.
We are finally reconstructing the complex cross-system dialogue that really defines human physiology. It's a convergence of spatial biology and genetic engineering that it opens doors we previously didn't even know existed.
It really does. So what does this all mean for you listening right now? Beyond just the incredible lab techniques and the molecular biology, I want you to consider the ultimate clinical horizon here.
The future application.
Right. Right now, if your heart's electrical system fails, the absolute gold standard of care is a surgical implant.
A traditional pacemaker.
Exactly. A surgeon cuts you open, places a titanium box with a lithium battery into your chest, and threads metal leads directly into your heart muscle. And look, it is a miracle of modern medicine, and it saves countless lives every single day.
Absolutely.
But wires snap, batteries die, and a metal box cannot feel your adrenaline spike when you're scared or naturally taper your heart rate when you fall deeply asleep.
Because the current technology is static. It forces a rhythm rather than actively participating in the body's natural autonomic feedback loop.
But looking at the blueprint laid out in this 2026 paper, what if the future of pacing doesn't require metal at all?
It's a profound thought.
What if a decade from now, your doctor just takes a swab from the inside of your cheek, reverts those skin cells back into stem cells, and grows a customized biological pacemaker, a perfect, living Sino matched exactly to your DNA?
No immune rejection.
Right. And what if they simply inject that microscopic tissue directly into your failing right atrium where it follows those chemical breadcrumbs, seamlessly wiring its own axons into your nervous system?
It becomes a part of you.
Exactly. No batteries to replace, no metal wires to break, just your own biology repaired, fully integrated, and beating in perfect sync with your nervous system.
That is the ultimate promise of this research.
It's unbelievable. Thank you so much for joining us on this deep dive today. Keep questioning the incredible biology inside your own chest, and we'll see you next time.